All right. Hi, everybody. Looks like we have 9 people here. Hi, I can see you, Kevin.
Hey, Carrie. How you doing? Hey, Julie.
Hello. Hi, Kevin.
How are
I'm
you?
Carrie. I'm the host of the meeting. Julie here will be our leader, our expert with the topic. We're going to wait just a couple minutes for people to get signed in and figure everything out.
Carrie, I can't see anyone because I have my
Okay,
my
I'll just take a
slideshow.
quick
But I have my chat box open and I could see you and me.
Okay. Yeah, and everybody, just so you know, you're all muted right now, but I'm pretty sure you can unmute yourselves as needed. Oh, hi, Eric.
Yes.
Hi, Margot. Hi, Irene.
Carrie.
It's great to see so many faces that we know. It looks like we still have, so we had 24 people sign up. And since this Zoom stuff is new to so many folks, I am going to wait a few minutes just to make sure people have time to figure it out. Looks like there's 2 more. I'm kind of backlit, but you can see me enough, I think. Maybe everybody, while you're waiting, get familiar with the chat feature if you haven't already. That's how we're going to communicate for some of this. Oh, wow. It looks like I'm not the only one who's outside. Looks like Margot is outside right now. That's so nice.
Carrie, I'm jealous of your backyard. It looks amazing.
That's really nice of you.
Beautiful.
Thank you, CNPS, Native Plant Society, for their plant sales. They've propagated a lot of my yard.
And you were saying earlier you had hummingbirds, right?
Yeah, that's because I have wonderful neighbors upstairs who feed them.
Oh, nice.
Yeah.
Awesome.
So I'm just going to wait 1 more minute, even though we don't have even half the people that registered, just because it's only an hour. These events are only an hour now, so we don't have too much time to waste. So we'll wait 1 more minute.
We're waiting. There's a podcast called Invisibilia, and today's or yesterday's, I'm not exactly sure when it was produced, but it featured Bernie Krause was the lead on it, and it was all about his work on the Natural Great Animal Orchestra. And it was very, very good. So if anyone gets a chance to listen to it, it's well worth it.
Go ahead and type that in the chat function, so the chat bar, so people can remember.
I actually have the link, but it's awfully long.
That's a really good podcast. I always love listening to them.
Me too.
They're great.
Well, everybody, since there's only 11 of us, I thought there would be 24, and that would be too many for everyone to be talking and videoing. But since there's this few of us, if anybody wants to, we can just unmute ourselves and have our video show. It's up to you. So I'll leave that up to you to decide. But I am going to get started. So thank you all so much for being at our very first virtual event with the Center for Environmental Inquiry at Sonoma State University. So this is Citizen Science, an intro to citizen science, and iNaturalist is the app that we are focusing on today. So I'm Carrie Winninger. I'm the outreach lead for the Center for Environmental Inquiry. And our public events are usually done outside, either at Osborne Preserve on Sonoma Mountain in Pengrove or at Galbraith Preserve, which is near Yorkville in Mendocino County. But we're all feeling the challenges of shelter in place, just like you surely are. And we're so happy to be able to reach out to people during this time and connect in new ways. So thanks for being here. But do please forgive any minor technical difficulties as we are navigating this for the first time. So before I let Julie take it away, I just want to tell you a little bit about what the Center for Environmental Inquiry is how it can be a resource to you, no matter if you're affiliated with SSU or not, whether you're a student, a parent, a government employee, an educator, a member of the public, or an organization that's in need of environmental solutions. So the center envisions a North Bay that's working together to find sustainable solutions. And it invites you to get environmentally ready with us. That's what our shirts say, if you could see that far. Get environmentally ready, environmental readiness for all. And we're doing that providing firsthand understanding of our connection with the environment and skill building experiences like today that result in sustainable solutions. So we're talking about helping people become aware, prepared, and engaged. And we're talking about all sectors of society. So there's a lot of ways to get involved. You can engage in research. We have a naturalist and land management training programs, internships and student jobs. There's events like these. We have data that you can access. You can lead or contribute to events or partner with us on projects. So lots of ways. If you want to get involved, just send me an email at the end of this or check out our website, cei.sonoma.edu. So you are important. That's what we want to get across. Everybody that attends these events and is involved with the center is an important part of tackling the greatest environmental challenges that humans have ever known. So thanks for being here. Today, we're going to focus on biodiversity and how we can harness technology to help contribute to science in a way that helps solve those environmental challenges. So let me just give you a bit of an overview. So this is going to be in the local nature format, which means it's going to combine a presentation with a local nature exploration. It's an hour-long event. After I talk for 2 more minutes, Julie will talk for about 25 for a PowerPoint presentation. After that, we'll give you some instructions on what to do, and then you'll go off for 15 minutes on your own to find your own piece of nature. Now, local nature means any area that you have access to during their shelter in place where wildlife or natural organisms might be. So this could be your backyard or front yard. It could literally be a view out your window if you're inside. Maybe there's a lot around the corner where some plants are growing where you walk your dog. Maybe even in your own house, because we've all had flies and ants in our kitchen. We didn't put those there, so that still counts as natural organisms. So we'll spend that 15 minutes exploring. This time using the iNaturalist app, and we'll come back for the last 10 or so minutes to discuss, ask questions, and then hopefully wrap up. So that's the format of the day. Before we begin, you might want to get out your iNaturalist app on your phone or your website if you can have a separate browser from your Zoom. That way you can follow along with what Julie's doing. You don't have to do that, but it might be helpful. Also, just some housekeeping. Like I said, I had muted you all, but feel free to unmute yourselves since there is only 13 of us now. So feel free to unmute yourselves. And I do see some people have joined since I started talking, so you'll just have to catch up. Welcome. I want to say a couple things about Julie's presentation. So she has a really cool system. So she has an asterisk in the top of some of her slides, and that means she's asking for your feedback. And we're going to ask for that feedback via the chat function. So make sure everybody knows how to use that chat bar and just send it to everybody. And so you'll see when that comes up, please answer that way. If you have any questions that come up during the event, I will try and monitor them, but we're not going to interrupt Julie to ask them. But feel free, and I can try and figure it out while we're going, or we can get to those at the very end. So if there are any other things Julie wants to say before about technical things, go ahead and say that. Otherwise, turn it over to her and have a nice talk, Julie.
No, thanks for the intro, Carrie. And I just was noticing 2 ants on my my computer monitor. So yes, definitely plenty of observations to be found today. Hello, everybody. My name is Julie Wittmann. I am Sonoma State University's Community Naturalist Program Coordinator, and I'm also a former SSU master's student and teaching credential student. I'm also a local biologist and California certified naturalist. Today I'm going to share with you very brief intro into citizen science and iNaturalist. So welcome here. It looks like we have one more person in the waiting room.
All right, I just admitted them.
So So the term citizen science can be called different things. People have called it public participation in scientific research, community science, crowdsourced science, volunteer monitoring, and I'm sure I'm missing a bunch of others. So what is citizen science? So in the chat box, please type in a few words about what you think citizen science means. So in the chat box, is everyone able to find it okay? Please share what you think citizen science means. All right. Excellent. Keep on contributing. So it sounds like if you're typing anything, and some of you already have, mentioning something about unpaid volunteers, making contributions to science as part of a collaborative project with professional scientists, then you are correct. So this can include, you know, related to, you know, project design or monitoring and data collection, or data interpretation or data verification. Good, thank you guys for contributing. Excellent. So many people are familiar with Audubon Society's Christmas bird count, conducted by volunteers since 1900. So it's been, you know, one of our longest citizen science projects we've had in North America, and it's considered probably one of the longest running citizen science projects. And so basically with the Christmas bird counts, an experienced birder leads a group of volunteers as they collect information about local populations of birds. And so this wildlife census informs bird conservation efforts and long-term species patterns. So as you'll see, there's an asterisk on this slide. So thanks for those of you who contributed with the last slide. So let me ask you, have you guys ever joined a Christmas bird count before? We have had them at Osborne Preserve before, but maybe you've joined another one, or maybe you've never joined one, and that's okay. So feel free to share in the chat box if you have been part of a citizen science project, specifically part of a Christmas bird count before. And feel free to share which one or what area. Christmas bird counts for kids, yep. Excellent. Thank you for sharing. Other citizen science projects folks may have heard about, or maybe not, would include the California Native Plant Society Rare Plant Treasure Hunt, which is a volunteer program that supports the conservation of our rare flora, our rare plants, by providing data to the California Native Plant Society Rare Plant Program and also to the California Department of Fish and Wildlife. So with this type of citizen science event, treasure hunters can join an organized rare plant search or learn how to plan their own trips by attending one of the training events. Whereas a BioBlitz, as you see also here on the screen, it's a biological inventory or census with the primary goal, main point is to obtain overall count of any biological organisms in a specific area. Could be organized by National Geographic or parks or other organizations. So that would be another example of a citizen science related event. So utilizing efforts of citizen scientists has many benefits. Research, for one, could never collect this much data for so long without the support of the general public. And it also, you know, involves free labor, right? So that's always a positive thing. However, there may be certain limits to what volunteers can be involved with. For example, private property considerations, or actually that could be a pro and con depending on if you're using a phone app, which we'll discuss later. But you know, or maybe there's concerns because there's special status species involved, and permits are required for research and handling of those species. Or maybe there's just, and this is kind of a big one, concerns with the accuracy of the volunteers and their data collection. So accuracy problems are often the first critique of citizen science data, and this has been assessed in numerous studies. Certain error may occur with certain projects or steps of projects, but the benefits of contribution from citizen scientists are generally viewed as useful. The big one would be the Christmas bird count, the longest ongoing bird census that, you know, in the previous slide that I had showed you a few slides ago that I showed you about, provided some amazing insight into, you know, long-term patterns of birds. So I just wanted to share briefly my example, my herpetofauna meaning amphibian and reptiles, my project that involved biological monitoring by volunteers. This occurred when I was a Sonoma State graduate student. It included a study comparing volunteers and more experienced herpetologists and various steps of finding and recording the various groups of amphibians and reptiles. And it was determined that volunteers were not very accurate at finding camouflage salamanders or photographing fast-moving lizards, but they were exceptionally good at finding and recording all species of medium and larger size salamanders, and also photographing certain groups of animals, specifically the salamander group, the snake group, and the frog group. And the volunteers or citizen scientists were amazing at recording a new species, specifically for pepperwood that was not on the preserve list before. It was a black salamander observed there. And so for the first time ever, you know, folks actually documented and recorded and observed a black salamander, which is, you know, added new onto the species list. So, and you could see the references slide at the end of this presentation for more details about my research and results if you're interested. But everything is also listed on my website, protecthabitat.com. So in the last few years, the field of citizen science has expanded more rapidly with the development of smartphones. Okay, so allowing more information, you know, that could be shared through digital media, and smartphones, which we all know have built-in GPS receivers. So volunteers or citizen scientists can readily provide geolocation information and timestamp information about species or even situations in real time. So there's a huge variety of citizen science apps now, and here on this slide I included just some examples. That would be the SODMAP Mobile, eBird, Project NOAA, iNaturalist, Globe at Night, which I just heard about on last week's Science Friday. They had SciStarter representative on, and it's a light pollution monitoring app, so that sounded kind of interesting. So there's something for everybody out there, whether you're interested in biology or light pollution, something for everybody. So in the chat box, as you see, there's an asterisk on this slide. If you have any citizen science apps on your phone, what do you have? iNaturalist, hopefully. So please feel free to share in the chat box what apps you have. iNaturalist, good, iNaturalist again, yay. Woo. And hi everyone, I'm finally seeing your names now. iNaturalist, great. Whoa, Ben, that's a lot. All right, Margaret. All right, so today we're gonna just focus on the iNaturalist app. Great, and which is a very popular biodiversity app. It's free, of course, as many of you realize. So the functionality of iNaturalist is akin to the Facebook of biology. where media evidence, typically photos, but it could be sound, and written exchanges can be shared with anyone in the world, whether they're a novice or an expert. iNaturalist is also used as an identification guide, an info guide, and an electronic journal, as you can see here on this slide. My life list, my username is Protect Habitat on iNaturalist, so that's my life list. I have lots of herbs, so as you can see, but iNaturalist, can include any biological organism, most commonly fungus, animals, and plants. So, this app documents things, living things, and so it doesn't document the lack of data, but it documents data. So an observation is a data point. You can't just say, oh, there was not something not here. So has crowdsource, the other important thing is it has a quality assurance directly built into the app. And this built-in function addresses that big issue I mentioned to you guys about, data accuracy, and the biggest frowned upon thing about citizen scientists. But the fact that, when 2/3 of the community agree on a particular species ID, then it becomes this so-called this green colored little research grade, which I'll show you which you'll see on the slide coming up soon. But so that just means 2/3 of the community agrees. So it's pretty, I'll mention a few things about the results of my research, but it's accurate. There's also other quality grades that your observation on iNaturalist could be. One is casual, which is not so good, missing some information, and then also needs ID. But research grade is the top-notch kind that you can get on iNaturalist. So the other thing I was going to mention here is my research resulted in 100% accuracy for all amphibians and reptiles, all the herp groups documented within my iNaturalist herp project. So once it's research grade, at least for herps, you can count on that that is actually what it is in reality. So a very, very accurate crowdsourcing identification going on with iNaturalist. So again, last slide of the references, you can get that website information. So why is photo quality important? First of all, it supports the identification of your observation and good quality data. So how should we photograph, for example, animals to support good quality data? So first of all, you want to make sure to include unique identifying features, and obviously that is and often can be species specific. For a lot of amphibians and reptiles, that typically includes the lateral or side view photo of an organism and the dorsal, the back top view of the organism. I could go on about different groups, but different, critters have different areas where the unique identifying features of the organism occurs. But with the top and the side view is always a good start. So you want to also consider animal movement. That could be species specific and or involve environmental conditions. So if it's a hot day and you want to try to document snakes, you're obviously going to have have your charged cell phone with the app ready on a lookout for snakes, because when you see them and it's a warm day, they're going to quickly go. So there's certain considerations when using smartphone apps to document specifically certain organisms like snakes on a hot day. There's also considerations relating to handling organisms. So you've got to have a caution for animals and human safety. So obviously rattlesnakes and alligator lizards bite, so you want to not be handling those guys. And then also there's, small, slender salamanders can desiccate really easily on a day other than a rainy day. So, you don't want to be handling those guys. The cauterization of a skink tail, as you see on the slide, there's that blue skink tail. Those fall off very quickly. So that's why I've never recommended that volunteers grab and handle skinks, because they lose their tail, and that's a source of energy for them. They need that. It takes energy for them to grow it back. So So there's also special status species guidelines to follow. There's obviously in this slide here, well, not obviously, but that's a California tiger salamander on the bottom left. So that is obviously not to be handled unless you have very specific permits to be handling and researching those. But of course, if you see an endangered species, surely you can take a photo. You just don't want to handle them. And then, of course, there's considerations relating to capture tools. When photographing organisms, I generally don't recommend that volunteers capture animals. You know, there's snake hooks or nets, but unless you're trained on how to do that to care for yourself and animals, it's generally not recommended. Scooping up small salamanders, again, you got to worry about them drying out if they're really small. You know, some folks I've seen naturalists will scoop up a salamander with wet leaves, but it's really important if you do want to do a pinky touch or do want to gently handle something that you have what's called nature clean hands. So we're in the world pandemic right now, so we might have hand sanitizer on our hands right now. That's definitely something we don't want to be, you know, putting on critters. So you want to make sure to have nature clean animal hands for animals, like wash your hands with clean water or rinse them off in a creek and rub them with mud or whatever you do, but nature clean hands, you know, if you do want to do a little bit of handling on certain species, that would be the best idea. My favorite topic, critter containers. I remember I used to go to the dollar store, but they have these great white critter containers. I really want to go to the dollar store. It's been quite a while now, but the white ones, they're $1 for like 5. They make for a great background for aquatic or terrestrial animals before taking a photo. And also even having, you know, some sort of scale with a white background, as you see with the slender salamander here, is always a good idea. So this is just a white sheet of paper with a photocopied ruler. You know, nobody likes pennies anymore, but they are good for one thing. They are good for scale in photographs. So that's also another way to get good support from the online community to get your stuff, your observations identified. So
Julie, real quick,
Yeah.
someone was having a hard time hearing you. Is there a way for you to be a little louder?
Sure.
Maybe closer to a mic, or I'm not sure.
Okay. Was it inconsistent audio?
No, it was just kind of quiet.
Oh, okay, interesting.
And
I'll
if
try to
everyone
speak
can
up.
make sure that their computer also has their audio turned up.
Okay, I'll try to speak up.
Thank you.
So again, quality important. Again, it supports the identification and good quality data. So how should we photograph plants to support good quality data? So my research determined that photo quality absolutely matters for the identification effort by the online community, especially for herps. You know, several photos of various plant parts are important for an iNaturalist observation. Typically the whole plant, you know, including the base of the plant or tree. When I say plant, encompasses trees and forbs. And then if there's reproductive parts, those are especially important to take photos of. The flower, we have a lot of wildflowers in bloom now. That's always the top-down photo of the flower is always a good idea. Any fruit the plant has is always a good idea. Obviously, we don't want to be handling poisonous plants. I had my fair share of poison oak and stinging nettle over the past 3 months, but we don't want to be handling it to try to, you know, get a better photo. Although some folks will brag they don't get that, but I'm so jealous. So also scale is important to a certain extent for plants, especially for those reproductive parts. And then it's important to have a good background, which typically is always naturally occurring. I think that at least the ones in these photos come out really well with the background. Where can you collect data? This is one of my favorite parts. So I took this photo outside of my bedroom window right here. It was an opportunistic observation. It was, I mean, I ID'd it as a Cooper's hawk, but I wasn't sure if it was, you know, the more common sharp-shinned hawk. So I just kind of just guessed. I thought, oh, Cooper's hawk? But thankfully, someone ID'd it for me and made it go to research. A couple people ID'd it and made it go to research grade. See, remember I mentioned the research grade, the strongest level of community support for a particular taxon. It's that on the upper right on the iNaturalist photo. This is just an iNaturalist screenshot for this observation that I took right outside of my window. So that was a research grade. So it was indeed a Cooper's hawk. Very cool for a residential area in Santa Rosa. Opportunistic observations can also occur at or near your house. As Carrie mentioned at the beginning, you can take a photo of a flea on your cat or dog, a spider in your house, and I'll also add an ant on your monitor. Right now I have a major ant problem in my house, including on my wall here. I just took these photos outside yesterday. The poppies, I have a bunch of poppies growing in my yard, and then this other nasty weed, like actually they're pretty easy to pull up right now. I've been pulling up this all week, so I'm hoping somebody will ID that for me online. So, you know, even though we're all sheltering in place, we can always find something. So another opportunistic observation includes this pallid bat at Osborne Preserve Ed Center during the daytime, during our community naturalist program. I also thought I would mention, this is a good time to mention that for the iNaturalist observations, if you want it ID'd or you want confirmation of your ID guess, is to put the best photo first, because that's what the online community sees. So there's two photos here. One has, and by the way, the bat did make it out of the exit. There's a bat up there on the exit sign. That's not the best one to use first. The best one is where it shows up the organism close. So you can clearly see the unique identifying features of the bat, the ears right there. That was a unique, you know, animals are not always the easiest ones to take photos of, but you can upload observations of the evidence of animals. So, you know, we're not including, you know, plastic pink flamingos on our front yards or potted plants or pets, but you can include, you know, evidence of animals or even evidence of plants. I'm sure plant folks have some ideas about that. So evidence of animals such as, you know, animal homes like this dusky-footed wood rat nest, stick nest here, observed at Turtle Pond and Osborne Preserve, one of SSU's preserves near campus. And then other evidence might include a feather. Again, these animals can be native or not native, just not artificial, you know, not or domestic. So this is a wild turkey. And the little bright pink introduction mark, it just means it does not naturally occur here, but it still exists in the wild. Other evidence of animals could be scat, another name for poop. Ant evidence, although maybe it'd be hard to determine which species of ant, it's still an observation, evidence of an animal. So in the chat box here, what other evidence of organisms can you think of other than what I mentioned, stick nest, feathers, and scat? What other evidence? So in the chat box right now, take a moment. There's an asterisk on the slide, so just saying, I'd love to hear from you. What other evidence of organisms can you think of? Holes in trees, tree galls, animal prints. And I'm going to read all this in more detail. I know it's kind of going quick on my little box here, but thank you for prints, bones. Excellent. Thank you guys for sharing. Keep going. Gnaw, antler rub, claw marks on trees. Great. So you can include dead or live observations, such as this dead California legged frog found at Osborne Preserve at the entrance after a really heavy rain a few years ago. So by the way, again, this is one of the animals that cannot be handled anyways. In California, it's a special status species, and by the way, it has a little red endangered symbol at the top. But you can include dead or live observation. And good, I'm glad to see you guys are still contributing to the other evidence of animals that you can include in iNaturalist observation. Smells, interesting, smells. Pellets from owls, owl pellets. Smells, boy, that would be really cool. So anyone can create a project. iNaturalist project, such as our Osborne Preserve project, is showing here. But anybody can create a project, and one of the benefits of a project is that you can actually include everything that's found in a certain area or between a certain group of folks, and then you can export the data, you know, and see in an Excel file, and which again has all the date and time info. So one thing to mention about projects is if, for example, you are at Osborne Preserve and you want to include your observation in there, you want to make sure you have to actually add it physically in, at least more recently, you have to actually manually add it. So it's an extra field, it's an extra step. Sometimes a little bit more challenging for newer folks to do, again, because it's just an extra step with the technology. But once it's in there, it's a good thing to have. So I also wanted to take this time to mention that Sonoma State does have 2 other iNaturalist projects. We have one for Galbraith Preserve, and then we have one for the main campus. It's called SSU Biodiversity. So in the chat box here, how many observations does Osborne Preserve have here? So how many observations do you see? So this is the kind of information you can find by looking within projects. So how many observations does Osborne have? And what is the species most observed using iNaturalist? So how many observations total are there? And what is the most common species observed? 1,656. If you said that, you are right. Excellent.
And
Great.
Julie,
And
I'm sorry to jump in, but we are running a little short on time. It's such a short hour. If you could
Yes.
hurry just a little bit.
yes. So here's the Osborne Preserve project. So in the chat box now, please share with me what plant species is most commonly observed at Osborne. What plant species is most commonly observed at Osborne? Yay, California Bay. I wonder why that is, Carrie. Okay, the other thing I was going to mention about projects is these were minks found at Kelly Pond, and it was actually documented using iNaturalist. So we just added a new species to our preserved species list because of the use of iNaturalist. So one thing I was going to mention, if you take the Taxa Challenge, it's called the Taxa Challenge, and it's on the references slide, which I'll show you in a little bit. It's on the resources slide, so you could learn about new species in your specific neighborhood, town, zip code. And these quizzes, I think there's 10 slides using random iNaturalist photos. Of course, they have varying photo quality, but you can choose to test yourself on plants, animals, or fungus. And then if you don't get 10 out of 10 on that little quiz, you could just blame it on low photograph quality. But feel free to take a look at the references or resources, excuse me, list at the end here. So I'm going to just quickly go through the interesting observations and iNat capabilities. Interesting observations can include individuals with unique features, such as my former high school student took this western fence lizard with a forked tail. She did that at Howard Park. Another one is this black morph, stumping the online iNaturalist community. Black morph of a California newt, which doesn't have the typical California lower light colored eyelid. So that was an interesting one. This was documented by Sonoma State student researcher with Gurman Lab at Pepperwood Preserve. And other iNat capabilities could be those such as the Herpetofauna project that I focused on, involving cover boards, and it used a research method and a structured project design. So this is just an example of a method-based biological study involving cover boards and scheduled survey for pre and post fires at Pepperwood and Osborne Preserve in Sonoma County. And so you could just see that the research grade southern alligator lizard with the burnt shedded skin immediately post-fire, one of the first surveys post-fire at the bottom right there. And finally, for iNat capabilities, plant seasonality and phenology are other options to take a look at. For iNat tech questions, you can, and this is on the resources slide, you can use the iNat forum to ask any questions about the details. The technology is constantly changing, but iNat tech questions are often posted to the iNat forum. And so once again, my name is Julie Wittmann, and there's my email here, and my iNaturalist username is Protect Habitat, my website, protecthabitat.com. And next coming up is, and you might want to get ready to take a screenshot of this. This is that resources slide that I referred to throughout the presentation. And the iNaturalist article with the results of the data of the citizen scientists using iNaturalist to document herps is here. The taxa challenge that I think you guys should take after this virtual event. This link can be found here. And the iNat forum link is here as well. And then a couple other citizen science resources, SciStarter and Sudden Oak Death links are here. And then, of course, and Carrie will mention more about this later, Sonoma State University Center for Environmental Inquiry calendar of event link. So you can see all our other events that are coming up. And then, of course, my website again, that has kind of the list of everything, including iNaturalist curriculum for teachers and naturalist educators. And if you have any questions about these or anything else, especially involving Sonoma State's program, of course, outreach lead, Carrie Winiger, email here. I know, Carrie, you mentioned your email at the beginning, so I just wanted to, if folks take a screenshot of this, then they'll have it for later. And now it's time, the fun part. We're all gonna go out and make an observation. We'll have 15 minutes to do that. Before you head out, please share your iNaturalist username here in the Zoom chat box. Mine is Protect Habitat, and Carrie's is Carrie Winiger. So when you go outside, and maybe some of you guys have already done an iNaturalist observation before, but if you haven't, the way to do that, to create an observation, you open up the app. First of all, you want to make sure your phone location service is on. And then you open up the app and you click observe, take photos of the organism, and then you hit next and then add into, and then what did you see? So you always want to make a best guess. You don't want to just leave it blank. You want to put in your best guess for the finest taxa level possible, even if it's just insect instead of, you know, red ant or whatever. So, and if you're back early, you can go to iNaturalist.org website and follow others that are listed here, and those folks who are adding their usernames. All you do is you go to community, people, enter the username and search box, and then you hit search and follow, and then you can follow others who are part of this event today. And if you still have more time, you can ID someone else's specimen that they just uploaded, and even more time than that, you could check out our SSU Preserve iNat projects. And I'm just going to keep this slide on while we head out. Carrie, is there anything you can add?
Yeah, I'm going to jump in. We're only going to take 10 minutes because we're running late and I want to have some Q&A. So that means be back at 2:53. So go out, make some observations, and please be back on time. I'm going to start right at 2:53. All right. And I'll stay here on chat if you have any questions, but I'm going to turn my, my screen off.
I'll get this ant on my computer.
I love it. There's an ant there. So I'm going to go in, Julie, and add all of these names and follow them. And if anyone hears me with questions, feel free in chat or turn your audio on and ask me. So Julie, I am typing somebody's name in to follow them. So I did what you said, we'll do community, go to people, type in their name. Now, how do I actually follow them? Where's the button to follow?
Oh, it's on their profile.
I see their observations.
Yeah, it should be on the top somewhere. Follow, next to their name or something.
I don't even see their name. That's what's so funny. Let's go back one. Okay, I see. It maybe because they don't have any observations yet.
Hmm.
Oh, I see. I clicked on observations instead of their profile. Okay.
Oh, okay.
Now I can say follow. Okay, great. Great. Now we'll just keep doing that. Tell me if you have anything you need from me too, as I'm adding to folks.
Gary and Julie, it's Margot, and I don't see how to get to community if I'm just working on the app.
Oh, from the phone itself? That's a good
Yes.
question. Do you know that, Julie?
Oh, I don't, I tell people to go to the website. I don't think there's a way to log in on the app. At least there wasn't within the last year that I know of.
Well, you can definitely get in. I mean, I can see all of my observations on the app.
Margot, you know, I actually posted, if you go to my website, protecthabitat.com, and I don't mind changing it for you, or you could change it. It actually just gives the instructions on how folks can join Sonoma State's Preserve iNat projects.
Margot, are you trying to follow people or are you trying to do a preserve project?
Without having to go, I mean, I'm out here without my computer.
Yeah.
Okay, I want to follow somebody else.
Margot, I'm gonna keep adding these people, and once I'm done, I'll look on my app to see if there's something to help you with, okay?
All right, we'll
I
talk
want to
about
get
it
everyone
afterwards
added
too.
first.
We can talk later.
Yeah. I might be done in a sec here. It's a good question, though. Someone else probably has that question.
Oh, I just saw your private message. Sorry.
That's okay. I thought it might actually get through to you since it was during the time where you were asking
There
everyone
was
else for
so
feedback.
many. Yeah, they were rolling through, and my window, my chat window is small because I was trying to keep it small.
Yeah, no, I totally get it. That's why I unmuted and told you out loud. Come on now. It doesn't want to go. Oh, please don't freeze on the internet. That would be a bad time to do that.
This would be a great day for Margot to share her bird citizen science stuff.
Because of the birds.
Yeah. Is Margot still here?
Yeah, I was on mute.
Oh, okay. Oh, I just didn't know if I answered your question okay about how to get the community actually using iNaturalist.
Well, if you have to use the website, I'm sure that I can
Yeah.
figure it out.
Yeah.
I find I keep expecting that the app has the same flexibility, and of course it has nowhere near the same.
No, I'll email you the link that actually has an instruction sheet for how, and you're welcome to use it. All you got to do is change Osborne to Galbraith.
Okay. Okay, great.
It's the one that we use to get folks signed on to iNaturalist for this event.
Okay. That'd be helpful.
Or actually, you could just go there right now. It's protecthabitat.com, and then it's just, if you scroll to the bottom, it just says programs, and it says Sonoma State, and you just click on the first link.
Well, I can't get there without losing you
Okay.
on Zoom.
So Carrie, I just want to be prepared for when you want me to get rid of this slide. So you want me to just unshare? Just tell me when to unshare.
Okay. I'm furiously trying to add everyone, but my computer is going slow, so I don't know if I'm going to get everybody in before
Yep.
the time's up.
I could scroll down if you need me to read some of these off.
No, it's more about the computer. I can read it quick enough. Actually, I think I might get there because I'm already following Margot and Nicolette. So now just to follow Ben.
Actually, and when folks get back, I imagine they'd want to. I wonder if we should compile this. They can enter, since we won't have a lot of time. Can we save this chat box and not close it down right away?
We sure can. Are you typing to everyone, Julie, like bye K Bruno? That was you.
By what?
Did you type in the chat box recently?
Yes, he said goodbye to us, so I just,
Great.
yeah.
Oh my gosh, Margot, I wasn't following you.
Really quick, I heard people talking about the app because I was on my phone at the same time. And I believe you can add the project. I'm using the phone app, so you can scroll all the way to the very bottom and it says add projects or add this to a project.
Yeah.
You just have to be following them. You probably have to do the following setup part online. So that way your phone, you can't probably add a project you're part of. You have to already have it kind of on your list, I think.
Yeah, so
it used to be, oh, okay, go ahead, Carrie.
When I take observations on my phone, I can immediately at that time put it into a project, but only if I've already added myself to the project. So yes, I think you have to add online.
Yeah, that's the same thing I have too. But I still don't know how to follow somebody, but.
Okay, Julie, help me find
I'll figure it out.
Julie, I need help real quick because it's going to start real soon. So I added everybody, but I want to make sure I know how to get back. So let's see if you still have that slide up. Okay, community, people.
Yep.
No, I don't want to add. I'm trying to find who I am following. So if I go to my profile, following, that should work. Oh, and it's so exciting. I'm seeing people add stuff. Okay, great. So it is time to come back. So I am going to minimize that, start on with my video again. All right, and then we'll see how many people have joined us. I see a few people back at their computers. I am going to assume they are all back because I said it was strict timing. So now let's make sure I have my screen shared the right way. All right, Julie, so now let's go ahead and switch to my screen.
I'm going to stop sharing. Do you want me to stop sharing the slide?
Yes, please.
Okay.
All right, now
Oh, now I get to see everyone.
Need to screen share myself. Yeah, and everyone's here. All right, let's see if this works. Can you guys see this, my iNaturalist account up here?
Yeah.
Yes.
Great. All right. So, Julie, go ahead and tell me what to do or have questions. If everyone wants to unmute themselves, feel free to ask questions at this point. We only have a few minutes, but I just have up here on the screen my account. I've now followed everybody. And so I've seen that you guys have been adding observations. So we have Laura here who just found a California poppy, which is fantastic. We have Nicolette who just added what appear to be mushrooms. Irene found 3 things in this short time.
Mm-hmm.
Kevin found 3, insects or arthropods, I should say. Derek has 2 plants. Ben has something. Your photo didn't work. We have a prickly pear. Oh, that's great. Daniel. A jumping spider, Margaret. Oh, great job, guys. That's awesome.
And it's really unknown why photos don't show up sometimes. It could be like a server area error. Were you able to get that uploaded, Ben?
I'm still working on it. I know sometimes if you don't say the species, there might be some limits. So I don't think I even put a species. I'm not sure if that affects it.
I would love to learn where everybody found these observations while sheltering in place.
Yeah, does someone want to share where they went? Just speak out. Or you can chat if you're shy.
I can say I went in my backyard and actually found some ladybugs. They're pretty cool.
That's great. I see
Hey,
someone
can we
else.
go down to hers, Carrie? Could you scroll down
Sure.
to Nicolette's?
Yeah, the mushroom was actually from a while ago, but it seemed like it had oddly added, like since 2019. So
Let's see, is there anything recent? It could just be my internet connection. I do see other people in the chat. They found them backyard, front yard. So good. I'm glad you guys have access to nature. That's really great that some people don't. Yep, I don't know why it's, oh, here's your observations, Nicolette. There we go.
yeah, that one. There you go. Yeah.
Wonderful. So Margot was mentioning during the break that there are different features on the app versus the website. So if there's something you're not seeing on your phone, do go ahead and explore the website. There's more there. Oh, good. She had a side and a top view. Very nice. Now I know you can identify ladybird beetles by the number of spots, so that might help.
I was just going to say, I think I'm going to take a guess on this one. 7-spotted ladybird.
Yeah.
Good guess.
Yeah, the Cosmella septum punctata, which is pretty common.
So, Carrie, let's see how quick this goes, because what I can do is I can make my guess.
Okay. All right.
Observation here.
Like suggest an identification.
Yeah. By the way, I think everyone in my neighborhood's working from home. My computer's been so slow. I'm going to suggest an identification, and let me do 7.
And this is the time for people to ask any questions that they might have.
And help ID. There's or confirm IDs of others.
Ben says, I'm so bad with plant identification. Well, this will help you get better. Wish I had a coyote to spot. Ugh, me too, Ben, me too.
Yeah, I found a rosette, which I really hope is not yellow star thistle. I don't think it is, but it's one of my I think it might be a dandelion. There's so many rosettes out nowadays, or at this time.
How do you get your observation in front of the most number of people, says Margot. Julie might have some tips on this for me. I've joined a whole lot of projects. And anytime I find an organism that fits one of those projects, I add it to that project. And then all the people in that project get a notification that there's something new. So for example, I've joined California beetles, I've joined roadkill, I've joined Marin County plants. And so not only am I making it open to all of iNaturalist, but those particular groups of people see my observations as highlighted. So there's more of a chance that they get them ID'd that way.
And I would just like to add, having a good quality photo will make folks interested in even looking at your observation. I'd say that's number one. Also, just making your best guess with the closest taxonomic group as possible, because some folks actually, and this is one thing I learned in my herp project, is folks really love IDing snakes, especially when compared to the other herp group. So if it's a snake, you're going to get 99% of the people making an effort on IDing it. It could be because generally the large size and the photos and it's pretty, and but in general, if you can ID it at least to a large group, then folks can choose their interest on iNaturalist, and then that'll pop up.
Thanks for joining, Margaret. I see you have to run. And I just wanted to show that, look, 2 people have already identified Nicolette's observation. So I do want to stop screen sharing because I want to, that did not work. Pause share, stop share. Because I did want to just say a couple things really quick before we take off. If you have to leave, of course, that's fine. But I just really want to thank you all for being like our test subjects. Like this was our first virtual event. So you guys really are helping us also figure out the best way to do this. Thank you, Ari. So the more of these we do, the more polished they'll get. We'll probably stick more to the time next time, for example. And Julie, thank you so much for being the first presenter. That is a hard thing to do. So there is a lot we have scheduled this spring. We have over 24 events coming up, just like from now until mid-June. So please check us out, cei.sonoma.edu/calendar. And we're still adding stuff. And it's really a wide range of topics, things that you in particular might be interested in since you came to this event. We have a whole citizen science primer. This is one out of the first 6 every Friday at 2 p.m. for the next 6 weeks. We'll be doing something cool. And we actually have several other presenters here. Kevin is presenting on dragonflies. Margot is presenting on birding apps. Ben is presenting on wildlife cameras. He's a different time and day, not Friday. But I'm sorry if I'm missing anyone else, but those are the people I see right now on my screen. So things to, oh, and I'm also presenting about sudden oak death, and that's also not on a Friday, but then you can actually use SodMap Mobile. And the power of that one is you get your trees tested for free. So it's partially an app and it's partially you actually getting mailed a packet, collecting leaves and sending it to UC Berkeley for sampling. So like it's a free service to you also. And same thing with the wildlife cameras. It's more than just about an app. There's actually a lot of instruction that goes on in it. So if you have any feedback for us, positive, negative, questions, comments, send them my way. And I will route them to who needs to get them. And I just want to say thank you again. Please let us know anything and everything that we can do to help you in this shelter-in-place journey to connect with nature. So thanks everybody so very much. All right, I'm going to stay on just a couple minutes in case anyone has any burning questions because I don't have to leave immediately, but otherwise feel free and take off.
I have a burning question.
Yes.
Which is, how do I find out, is there a place on iNaturalist that has like a list of the endangered species?
Julie, do you know anything about that? Like maybe there's a project for endangered species or something?
Well, okay, so the iNaturalist way of saying something is a special status species is a bright pink exclamation mark. As you saw with that California red-legged frog observation that I had a screenshot showing up. However, because iNaturalist is like a global biodiversity program and app, they don't really have, you know, the specific, oh, this is, you know, federally endangered in, you know, California or whatever it is for the different states or for the different countries. So it's kind of just general. The ones that I've seen them use are endangered and vulnerable, and you could probably find some more. I haven't seen a way to actually, and feel free if someone else knows any different, but I don't think you could actually search for observations that way in iNaturalist, but you can look under species and see whether it has that endangered or vulnerable.
I thought of something else, Julie. I know there's been some talk. I don't know if iNaturalist has done anything about it, but they've obscured certain observations because they don't want people to know exactly where endangered species are because they don't want them to go pick them or hunt them.
Yes.
Oh, yeah. Mm-hmm.
Yeah, like for example, the California tiger salamander, you know, there's a much wider radius of the GPS location that is being shown by the public. And because I have tiger salamander observations on there, just that I use for work and I keep my own records, but it's the exact specific location is obscured. And I have people actually contacting me, where did you see this exactly? And I don't tell them because I don't want people to start, you know, collecting them, but you know, there is concern that, you know, this can be used to hurt organisms. So that's why they have that infrastructure in place online to where species that are endangered or vulnerable tend to have more of a like obscured specific GPS location.
I just Google searched while I was at it and I found, you know, I think there are projects, right? So there's one that's called Endangered Species of Ohio. Someone started that list. So there may be things like that you can search for, but they're not official, I think, in the way you're looking for.
Right. No, and actually I see now, like the words threatened or vulnerable or, you know, endangered seem to come up with various lists. And I do see the grids for those are much bigger. It's like in this region somewhere, or this was spotted, which is great. I of course support that 100%. I just, since I'm a space scientist, so but we do some education in this area with other experts, such as yourselves, in fact. But I just don't like. I don't want to come across some species. I generally don't like to pick up things either. So it's not going to be a problem. But I don't. I just want to make sure my kids don't threaten any endangered species, because I would have no idea. Like, I didn't know that salamander was threatened. So good for me to know. Great.
Thank you for being careful.
Thank you.
Everyone else just hanging out for fun, having a Zoom party with us.
Carrie, I wanted to mention, I reminded folks about the taxa challenge that uses the iNaturalist photos. I just saw it on Reddit. Somebody had created it using iNaturalist photos. So it's the taxa challenge. If you look in the chat box, something for folks to do when they leave here.
I want to do it to myself.
The only thing I would recommend to the person who created this, which is really an awesome thing, he should just see if there's a way to only use research grade photos, because sometimes it comes up with like goose gut spilled out or whatever, you know, like, oh, how do you ID that? But the taxa challenge, it's a very
It looks like Ben also just found something about threatened species. Do you want to share that, Ben? Oh my gosh, I have an ant on my ring right now. See? Wildlife everywhere.
Oh. So under any project, I think you should be able to go to observations, and then you can, there's like all the filters you can have. And so they have filters for like different types of species. And I believe somewhere there's a checkbox for threatened. There's also checkboxes for like cultivated and stuff like that. And I just looked at it quickly, so I'm not sure.
Global projects. So just look.
Oh, sorry. Sorry. Yeah, I just started with the global ones, but I think you should probably be able to go by project, because it's just part of the filters. So you can, it's part of the filters. It's like 30 different things you can select, and I think there's one checkbox for threatened. I have to go to, let me see if I can go to Sonoma State.
Well, you can check by captive. Vulnerable is not on here. But you could do by wild. I mean, if you go into that filter, like go under observations and then filter on the little button on the top right, you could show just wild, just captive. You know, you can click on many of these little checkboxes.
I see what he means. I see the threatened button.
Yeah, there's only one for threatened. There's not all the other categories of endangerment, unfortunately. Or I don't think there are, but I haven't used it that much. I just noticed it.
And this is where some folks can just go under categories. Like, let's say I only want to ID snakes, and I just go category and I click on the snake, and I can, you know, ID as much as I want, you know.
Wow, that actually might be really helpful for Laura, because it is the whole world observations that are threatened, and you can select things in that way. That's fascinating. Thanks, Ben.
Yeah, I see that now. That's really, really cool. Well, thank you again for for this hour. That was very fun. Thank
you so much for joining us.
Organized it.
Yeah, we're all here at home, but we'll still connect to nature and working, and I'm having a good time. Thank you, everyone, for joining me in my backyard.
It's great.
Awesome.
It was great.
Where are you, Margot?
Bob's on there too. Bob, can you unmute your video?
I can unmute his phone, but his video is already on, so it must be an internet issue.
Oh.
Oh, okay.
Maybe he's finding lots of stuff, huh, out there?
Yeah.
Well, all you guys got to do is go to my kitchen. I have so many ants right now.
Where's the most unusual place you guys have all found an observation?
I've really done very little of it, so I don't have an impressive place yet.
Nicolette, what about you? I think I've done a few at Galbraith, which has been pretty fun. Yeah. But
well, my one, since it's only people we know now, but I think this goes on the recording, so oh well. Is I had construction going on in my apartment, and they tore out a piece of the bathroom wall, and inside was a slime mold.
Oh, that's
Slime molds are not the same as regular molds.
That's
Slime
true.
molds are incredible and really cool. And I
And
was
that's-
so excited to find it. I was like, oh, I'm raising a slime mold in my bathroom.
a good thing.
Are they dangerous? Are they toxic? I don't know much about.
It's like a
Slime molds.
totally
No.
different category of organism. So it's like
Oh, okay.
a water mold, a slime mold, and a mold on your bread, like are like
completely different.
totally separate, you know?
Oh, cool.
Like so they're different and it's adorable. And they're individuals, but they create colonies and then they move together as one mind. You can go look up online slime mold mazes. They can actually navigate mazes. They're pretty crazy cool.
Oh, wow.
There might be some sort of intelligence with slime molds that we just don't comprehend that is really advanced. Like I kind of think of them as little aliens.
I've read that
Yeah.
there's supposed to be sort of like a hive intelligence or colony intelligence that they're communicating at some level. Very cool.
That's my weird one.
I
Well, actually,
have times
yeah.
I had times I wish I had iNaturalist, but it takes time to get it out and then to tag it. But I've had possums and raccoons in my backyard, and I've had not confrontations because I stay far away, but I've had visitations from them, and I should have taken photos with iNaturalist. So I definitely want to I wish there was like a way to just have a camera on all the time and like push a button somewhere. Like if it's on a smartphone, you just have a camera on your head all the time, ready for nature. But that might be a little crazy. But
like in
that
the
would be
future,
cool
when we all have like cameras in our eyes and it records the
Oh,
last
yeah.
10 seconds of your life,
Hi,
you can just
Bob.
say, rewind 10 seconds.
Hi.
that would be
Talking
cool.
about
Yeah, there's-
post-pandemic technology here, right, Ben?